So hi everybody, it is time to start. Welcome to the Office of Information Management and Analysis monthly brown bag series. The webinar will be strictly a 30-minute event for both presentation and questions and answers. Please everybody mute yourself because we couldn't manage to unmute the other participants, so we don't want to hear much of the background noises. Please note that. So today's webinar is titled An Introduction to eDNA for Bioassessment. The presenter is Dr. Dovi Kasim. Dovi specializes in studying the population community ecology of pelagic fish species. He combines the use of molecular telemetry and quantitative tools to study species that are traditionally data poor and have cryptic life histories. His PhD dissertation from the joint doctoral program in ecology in between San Diego State University and UC Davis focused on using both genetics and satellite tagging to understand population connectivity into highly migratory shark species. As a joint postdoctoral researcher between SWRCB and NOAA Southwest of Fisheries Science Center, his current research focuses on using high throughput genetic identification of planktonic fish stages to understand human impacts on pelagic fish communities. He also participates in multiple community outreach and educational programs to bring marine ecology to local communities. So with this brief introduction, I will pass the phone line to Dovi. So thank you very much, Dovi, for doing this presentation. You can go ahead and start your presentation.
All right, thank you. I just want to check in to make sure the microphone is working.
Yeah, it's working well here.
Wonderful, thank you. So first off, I want to thank everybody for either turning up or signing in and providing your lunch hour to help learning about eDNA. And of course, I want to thank the Water Board's Office for Information Management and Analysis for allowing me to present. And I'm hoping that by the end of this, everyone here will be just as excited as I am about the applications of molecular tools to help support bioassessment. So as many of you know, probably even better than I do, bioassessment monitoring can be challenging. There's many different species that people are trying to observe. Many of those species can be rare or cryptic, particularly species that are threatened or endangered, as well as those that are invasive. Those are some of the ones we want to be able to identify the most, can be the most difficult to actually identify or find in the field. And detectability will vary across different species, taxa, and habitat, as well as life cycle. So you can tell that it can be quite difficult. And
hi,
just
how you doing?
As an aside, I just want to remind people to mute their microphones unless they want to co-lead the webinar with me, which I would appreciate. Thanks.
Okay, great.
After recognizing how difficult those can be, recognizing that eDNA is a tool that might be able to help us address some of those issues. In many cases, it can provide better detections of some of these species, a fast turnaround time, and can be reasonably inexpensive to process. But it's also important to highlight here that due to its high sensitivity, it means these analyses can be easily affected by contamination. So it's important that if these are going to be applied, they're applied carefully. Okay, so what is eDNA? If I were to ask any of you if there were a dog in this car, chances are most of you would be able to correctly identify that the answer is yes, there is in fact a dog in this car. But were this dog to walk out of the car or move to the back seat, and I would ask you, has there been a dog in this system? You might note that there are still some clues to lead us to assess that there has in fact been a dog in this car. Similarly, other organisms will lead similar clues that they exist in their ecosystem, even if we're not able to identify them directly. So, for instance, if we have this aquatic or marine ecosystem, there are organisms found within this system. These organisms are going to shed their DNA in cells in this water. So even if these organisms were not to be directly obvious to an observer, their DNA is still found in that system. And if we were to collect water from that system and amplify the DNA from within that water, we would be able to identify that those organisms have been in this system. So what does the E in eDNA stand for? It's most commonly defined as environmental. So this is environmental DNA. You'll sometimes see it written as extracellular DNA, although I prefer environmental because of this next point. eDNA is comprised of multiple possible sources. It could be shed cells, so the entire cell, let's say skin cells of an organism, get sloughed off. It could be free DNA that's not found within a cell, and it could be cells and other free DNA that come out of an organism's waste. So when they excrete waste into the environment, they also excrete cells. And our ability to assess eDNA is thanks to a molecular revolution that has basically changed a lot of the technologies available for us to study DNA in the environment, and we're kind of riding on the coattails of biomedical applications. But these tools are now becoming more and more available to us, and this revolution has allowed us to address questions that may have been impractical or impossible to try and answer, just several years ago. This here is one example of a study that has demonstrated the ability of eDNA to be able to identify organisms within the environment, and also not just be able to identify it, but more reliably identify organisms in an environment than traditional observational methods. And as this tool becomes more commonplace, we're finding more and more such studies demonstrating the efficacy of eDNA as a tool for biomonitoring. And additionally, using eDNA allows us to detect this presence without having to disturb organisms. And this is particularly important if you're studying threatened or endangered organisms that capture could potentially damage them or remove them from the population. By collecting water samples and just assessing the presence by looking at the detection of eDNA in that system, you don't actually have to handle those organisms and therefore have those negative impacts. As well as potentially this does not create trap shyness. So collecting eDNA on one visit does not make it less likely to collect eDNA the next time you sample that system. Also, there are now, as this tool again becomes more mature, we're finding guides and protocols to help make sure that sampling is standard for eDNA within these systems, allowing us to compare studies among different systems or among different regions within a system. And I'm not going to get into the specifics of this of these protocols. However, I'll point out that they don't require a tremendous amount of equipment to do. You can easily get this equipment into the field, collect your samples, and get back to the lab for analysis. It's relatively quick. It's relatively inexpensive to do the sampling. If anyone has any questions about this, hopefully we'll have time to talk about that in more detail at the end. But I will point out here that it is really important that the proper care is taken while collecting samples in the field to avoid contamination. This includes wearing gloves, making sure to change and sterilize gear between samples and particularly between regions as you move, and handling both the equipment and the samples with care, because any contamination during sampling will lead to erroneous conclusions further down the line, and it will negate all the great work you can possibly do using this tool. Okay, so there are multiple potential questions of interest that people might be interested in answering with eDNA, including identifying rare protected species, identifying invasive species, hopefully early enough to try stem those invasions. Perhaps people are interested in characterizing the diversity of an entire community, or beyond the diversity, actually identifying what species or what taxa are found within any given system, as well as potentially identifying genetic functional groups within a system. And depending on what question you're interested in answering, there are different genetic tools available to try to address those. So kind of as a general guideline, I'm going to talk about two of these tools. There's species-specific primers, which I'll cover in a second, which are really good for just identifying whether a given system is a given species is present within your system. And then the targeted gene approach, which will allow you to look at diversity of multiple different organisms and multiple different taxa within a system, as well as shotgun sequencing, which I'm not going to cover during this webinar. So species-specific approach, what that entails is designing a primer. So a primer is a short piece, a oligonucleotide, so a short region of genetic information that will amplify and only work if a species that has its corresponding sequence is available and present in a system. So what species-specific approach will allow you to do is target one species at a time. It does not require actually having to sequence that DNA. So you're actually just doing what's called quantitative PCR, which I'll explain in a second, which actually helps us be relatively inexpensive as well as relatively quick. You can get results pretty quickly. And it also has an additional benefit of allowing us to quantify the abundance of gene copies within a sample. That's not necessarily the same as the abundance of individuals in the system, but we're currently trying to find ways to normalize and try and move from gene copy to potentially numbers of individuals within a system. So the way this works is, let's say we have this this river system we have here, and we have midges present in the system. You'll see in the bottom right of the screen, you have an area of high density. In the top left of that pool, you might have an area of medium density, and towards the left, you have a region of lower density. So we could go collect water samples from each of those regions, as well as water samples from an area that we know doesn't have these organisms, so a negative control, and samples from an organism itself, which will have a known density of organisms and hopefully gene copies. We then take those samples back to the lab, and we can run them on what's called the qPCR, quantitative PCR. And what we'll find, if you see here, what we have on the x-axis is number of cycles. So cycles are the number of times you do a PCR. Each time you do a PCR, you're amplifying, so you're copying the number of gene copies you have available to you. And on the y-axis, you have fluorescence, which basically gives you an idea of how many of those gene copies are available. And the blue line there is an arbitrary threshold that we say, if we have enough gene copies that we pass that blue threshold, we can identify that these organisms are in the system. So if you see that line that's sitting around 0 in this figure, that's our sample as our negative control. At no point do we cross that blue line. So we say there's no DNA copies in that sample. If we then run our reference sample, we see that after around 18 PCR cycles, it crosses that threshold, and we can say positively that this organism was in that system. If we run the sample that came from the low density, we'll say it does eventually cross that blue threshold. It takes a lot of cycles to get there. The medium density sample will cross that threshold after fewer cycles, and the high density even after fewer cycles. But eventually they all do cross that threshold, so we can say that that species was in fact present in the system. And we can then use the gene copy abundance relative to our reference sample to try to estimate the number of gene copies that were available. We're going to now show, compare that to a barcoding approach. A barcoding approach does require sequencing to try to identify what's in a sample. So what we have here is, let's say can everyone hear me?
Yes.
Okay, so if we have a barcode, so we have a sample here, we know that there's organisms there. They might be difficult to identify morphologically, which is how we would traditionally try to identify what's in there. So instead, we'll take a molecular approach. We'll take one individual at a time, extract its DNA, sequence that barcoding region within that DNA, compare it to a reference library, match up what sequence matches to what reference sample, and then we can identify which species that individual sample was. However, we recognize that there are a lot of samples in here, so we need to do this for each one of these individuals at a time. And what gene region that we use as our barcode region will depend on the taxa we're looking at. So again, if this is something you're interested in doing down the line, I can talk with any of you in the future about what region might be the most useful for your particular study. And this has been really great. It's thorough. It's shown to be both accurate and reproducible. You don't have to have the same sort of taxonomy expertise that you need for morphological ID, but it can be expensive and time intensive as you have to go through each individual in your sample one at a time. If we were to compare this instead to metabarcoding, whereas instead of extracting DNA from a single individual at one time, we extract the DNA from all of the individuals in our sample and use this new high-throughput sequencing approach to identify all of those at one time, we can then actually speed up that process significantly. But of course, in order to know that works, we have to validate that this tool does in fact work in the way that we think it does. And if we were to compare this now to eDNA, where we have this sample, each of these things has their DNA, but they're no longer there. It's essentially the same as this metabarcoding approach, although instead of collecting the organisms, you just have a water sample or a soil sample that contains the DNA of these organisms. And again, we can sequence all that DNA using high-throughput sequencing approaches and identify everything that was in that sample in one go. Okay? But there are special considerations for eDNA. The first one is the shedding rate. So how often does an organism shed cells or DNA into the system? And that can vary among different kinds of organisms, as well as within different life stages, so across ontogeny of each of these organisms. So a cyst or an egg might shed less cells or fewer cells than an adult or juvenile. So that has to be taken into consideration when looking at eDNA within a system. We also need to consider the persistence. So how long will that eDNA exist in a system unattached to the organism? And that's going to vary depending on environmental conditions. So colder water tends to allow for longer persistence times than warmer water. Exposure to sunlight, UV radiation, is notorious for damaging DNA. So the lower the exposure to sunlight, the longer the DNA will exist within the system. We also need to consider biological conditions. So certain organisms and microorganisms will actually consume DNA in the water column. So the higher the presence of those potential DNA or cell consumers, the lower the less amount of time the DNA will exist within a system. And then we also have to consider current or flow or wind, as those will move DNA through a system, and we need to account for that when trying to study or collect samples from within a system. Fortunately, persistence and transport is something that can be tested for, and this needs to be done in each system separately. So you can, do spiking studies and see how far down a river you're able to detect DNA, or put DNA in a bucket with similar light conditions and see how long after a spike within that bucket you're still able to detect that DNA or that signal. So you can actually test for that beforehand, which is a good thing to do in any sort of study, and make sure that you account for those potential issues in your system or in your studies. You also have to consider the fact that contamination is a major issue for eDNA, because we're looking at low densities of eDNA, much lower than if we extract DNA from the tissues of the single individual. And often, because this DNA has been sitting in the water, it's been exposed to sunlight and other organisms, it can be in degraded conditions. So you have to take a lot of care that you're not accidentally contaminating your sample with splashing one sample next to another, or not wearing gloves, or cleaning the equipment appropriately. So eDNA can tell us a lot of things. It can tell us whether a recent target species or a target species of interest is present within a system. If you're using the qPCR approach, it can tell us the amount of eDNA within a sample, and hopefully we'll be able to correlate that with the population densities. We can use eDNA to identify the genetics of pathogens and potential hybridizers, although it's not a tool that can give us everything. It doesn't give us population size, although we do think we'll get closer to being able to estimate that. It doesn't give us the age structure of those populations or reproductive status. So it doesn't necessarily replace traditional monitoring, but it does complement it in such a way that it allows us to provide results on presence and hopefully some sort of idea on abundance in a reasonably cheap and quick manner. But of course, it needs to be validated. So I'm just going to go through a couple studies that are being done now with SWRCB in conjunction with some of their member agencies. We're working on a project now trying to validate the use of metabarcoding for ichthyoplankton. I apologize, it's moving forward on its own, so I apologize for that. But so what we're doing is we're collecting ichthyoplankton. So samples of planktonic fish, which are difficult to ID. We're trying to ID them traditionally under the microscope, as well as using this metabarcoding approach to ID them, and making sure that we actually ID the same species within those same samples to demonstrate that this metabarcoding approach does in fact give us data that's congruent and similar to traditional approaches. Also, this is work led by Suzanne Thoreau. So excuse me, I need to. SWRCB, looking at can we use eDNA to identify algae in a system and therefore develop an index for looking at ecosystem health by looking at algae within a system. Algae is traditionally very difficult to identify, and therefore using molecular approaches has a lot of promise to be able to identify algae and develop indices based off of those algae in the future. We're also working with the San Diego Water Board as well as several others to use eDNA. So this is a project that's just getting started. To use eDNA to look for the presence of invasive species within these systems, as well as identifying threatened and endangered species. Again, these are things that can be very difficult to ID using traditional methods because these are things that are rare, potentially hidden and cryptic within those systems. So hopefully what I'm able to demonstrate to you guys is that this eDNA approach has some applications to try fix some issues in traditional biomonitoring, including enabling us to increase the scale and capacity by providing a cheap and quick way to assess a system. It also doesn't require trained taxonomists in the same way that traditional approaches do. So as we are losing the ability to train people to be robust, thorough taxonomists, this approach can help us identify species. And again, it's in a reproducible manner between labs, help us ID difficult to ID organisms, and also in areas that might be difficult to sample with traditional methods. And many of you might have a question then, since we're doing all these validations, is how mature is the science? How ready are we to apply these tools? And I like to think that we're on the precipice of this, where these tools exist. We're doing a lot of the validations. When I say we, I mean the general scientific community, some of us at SWRCB and NOAA being among those. But we're doing some of these validations at the moment. Within the next couple of years, these tools should be validated and ready to go and available for biomonitoring, in some cases much sooner than that. And again, using these tools, there's a lot of different possible tools, whether you use the species-specific approach or the targeted gene approach. Which one you use depends on what your question is and the appropriate use of the appropriate tool given that question. Answering these questions about the health of ecosystems is very complex. So these tools are best used in conjunction with other tools, including traditional tools, to get a better picture of what the entire system looks like. But hopefully, in this short amount of time, I've been able to at least convince you that these are useful tools that can be applied along with traditional monitoring. And with that, I think I finished with enough time to allow for questions.
Does anybody have any questions?
Hi there, this is Chelsea Tellers from City of Davis. Can you hear me?
Yes.
Hi, it's actually not more of a question, but I appreciate that you're putting this forward. The Central Valley Clean Water Association is working with the Regional Water Board Central Valley, and we've done the eDNA study of freshwater mussel from the Pitt River and Soundfire water. in conjunction with the UC Davis lab, and it works. We are so thank you for putting this forward. We've learned a lot of things, and we're still in the process of doing more testing this spring.
Wonderful, that's great. And yeah, I think that, you know, there are many people working on doing some of these validation studies, including obviously you guys with UC Davis. And I think that the more these studies are done, the more we learn how useful these tools really are, as well as what some of the caveats are about using those. But I mean, I do see these tools being applied more and more and more. The key now is to make sure that we apply them appropriately. So the fact that these tests are being done is definitely a first step in appropriate application.
I'm Davi.
Yes.
This is Calvin. I had a question. Do you have a example of what the data looks like right now? Just curious to see what it would look like. Like, is it a list of species with like a check mark of present or absence, or is it more complex than that?
That's an excellent question. It depends. It depends on which approach you're looking at. So if you're using the species-specific approach, you're going to be looking. The data essentially will look like in each sample what the estimated number of gene copies are, or how many cycles were required before we crossed our threshold. And so that's going to look like one species at a time. Of course, you can run multiple different species-specific primers on a given sample. If you're looking at like the barcoding or meta-barcoding approach to eDNA, there's multiple different bioinformatic pipelines that are all going to end up giving you data that looks slightly different, but essentially each one's going to give you a data sheet that'll give you what species were present in your system, as well as the number of gene copies within that as well. But whether or not we can use that number of gene copies in the same way we can use the qPCR data, that's still something we're working on. And again, when I say we, I say the general scientific community. But again, it depends on which bioinformatic pipeline you're looking at, but most of them will include blasting your sequences against some sort of reference library. So it'll ID which species are there and the number of gene copies that you have within that sample.
Okay, thank you.
And again, if anyone, after this wants to email me with more questions, and I can try to potentially share some data sheets that may or may not look great, but I'm happy to talk about that further with people.
Can I chime in with another question? Is there time?
Sure.
Hi, it's Lisa Thompson at Sacramento Regional County Sanitation District. I've been involved in the study that Josie Tellers mentioned. And Dovi, thanks very much for your talk. This is a really wonderful overview and lots of great reminders about the opportunities for using the new eDNA techniques. The question I had, and this kind of came out of the freshwater mussel study, was if you don't find an organism present in your sample, how much effort do you need to expend to make you believe that maybe it's not present? In other words, trying to prove absence, especially with rare species, because it would be worrisome to write a species off when it was just so rare that you didn't detect it. I just wondered if you had examples of where anybody had tried to figure out what a threshold was in other systems and other species.
Yeah, I agree. Sure, that's a great question, and it's true. The data that we provide as a general rule with this tool is a presence-only data set, not a presence-absence data set. I know that with the ichthyoplankton, one thing that I'm doing with species that are of particular importance, I'm running basically mock communities with different densities of those particular species. So for me, that might include sardine or anchovy, which are particularly interesting for fisheries management, and saying at what density in my sample do I reliably detect it present every time versus at what densities do I detect it some of the time but miss it some of the time, to be able to say that if I don't see it the density of this species is below this threshold. And so, similar things can be done in other studies with spiking experiments to see how often do you detect it given that it is in fact present. And that is going to depend on the system. It's going to depend on water flow. It's going to depend on persistence given temperature and other sorts of organisms in the system. But it is a great thing to consider and test for in each system.
Thanks very much. That was super. Thanks.
All right, we're about out of time. Like Bobby said, if you have more questions, email him. Thanks, everybody, for participating. We'll see you next time.
Thank you, everybody.
Thank you.
Thanks very much.